we'll start with our impact slide. If you don't remember anything about that particular bacteria, you at least will get a little bit of maybe 5% or 10% of information. Bacteroides. Bacteroides get used to the term. You will see that a lot in hospitals. And be prepared for tomorrow's exam in terms of aerobic, anaerobic, these type of, pay particular attention to that. Gram-negative, gram-positive. This is an anaerobic gram-negative bacteria. So one of the important things that we want you to know, the major classification, what are gram-positive bacteria and gram-negative bacteria. And if you don't know, basically, there's a blunder. There's a very serious thing. So please make sure that you know very well. Impact slide. So you can see from here, number one is a polymicrobial infection. Remember, we teach you bacteria separately, but usually they come in group and cause problem. Does involve Bacteroides fragilis and other anaerobes. So anaerobes can be that nasty because of the tissue injury, because of the infection. This infection started in scrotum over here for this male and rapidly spread up. And you can see how rapid that spread is for this nasty bacteria. And you really have to act very fast, very fast. You have to do a gram staining, get it over with, and start the therapy. And because if you're late, then again, the bacteria is going to release toxins. Bacteria is going to release, in this case, it's more of a tissue damage. And tissue damage is a major problem. It's chewing up fat, it's chewing up muscles, it's chewing up fascia, and again, serious skin infection. Of all the group that we have within this genus, especially Bacteroides fragilis is basically a pleomorphic, but as I said, that sometime if you look at the gram staining, you'll see a varied picture. Some of them are cocci, some of them are rods, and it makes you think that there are many different bacteria. There could be, but pleomorphic means that they come in more than one form. So not a very strict staph kind of a thing. That should be a cocci. Had it been the case, we would have put that as their name, Staphylococcus. We showed their cocci. Streptococcus, they are cocci. Right? But when we don't do that, they're not cocci. I mean, it's as simple as that. Typical gram-negative cell wall structure. And I think, again, difference between gram-positive and gram-negative bacteria, we want you to know. At the same time, also keep in mind that in order for you to understand the cell wall, we do want you to know the structure of mycobacterium as well, and as a differential diagnosis, the structure of mycoplasma as well. And that's again in the lecture that we already discussed. Gram-negative bacteria can be as nasty as they can get, and you have a good reason, because they have outer membrane, they have periplasmic space, and then they have LPS, right? They do have a peptidoglycan layer, but that's thin. They have a polysaccharide capsule. Major component of the cell wall is lipopolysaccharide. And if you remember me saying, please know the difference between lipooligosaccharide and lipopolysaccharide, and which part of the lipopolysaccharide acts as an endotoxin. Some of the bacteria may have a weak endotoxin as compared to some bacteria that may have a strong endotoxin. And if you compare that, the LPS molecule in Fusobacterium and other aerobic gram-negative, that's in contrast to that. Bacteroides has little or no endotoxin activity. That's what I wanted to kind of emphasize to you. Though it does have a LPS, and if you pay attention to three structures of LPS structure, and one of them basically rendered that endotoxin activity, especially the lipid A component, because the reason is that it lacks phosphate group on glucosamine residues. So this is important for us to know in terms of structure, because also keep in mind one of my favorite questions is I want to ask you, what are the characteristics or physical effect of endotoxin? What is it that endotoxin causes? It causes shock, death, easy for you to know. but also keep in mind what other thing it may cause. Disseminated intravesical coagulation, for example. And then again, fever. Because why is it called fever? Because they are pyrogenic activity. And if it doesn't have an endotoxin activity, there will be less fever or no fever. Many a time, classical signs of inflammation has fever. And many bacteria like Bacteroides may not have fever because their LPS has little or no endotoxin activity. And for a bacteria to have an endotoxin activity, they have to have a strong LPS because it's a pyrogen. Okay, now if you look at pathogenesis, anaerobic, colonize the human body, and relatively few are responsible for causing disease. And as in the impact slide, you pretty much saw it was more to do with abdomen. So keep that in your mind. Intra-abdominal infections are associated with the Phagocytosis. So that again is an impact thing. And it is a minor member of gastrointestinal flora. So if you look at the percentages, some of them have more in terms of numbers as compared to some which may be minor. Why do they have such a nasty virulence factor? Because they cause the tissue damage. Also know the difference between bacteria colonizing and causing disease. They can colonize and may not cause disease. That's another important concept. So the virulence that you see in this and other pathogenic is coming from virulence factor. One of the virulent factor is adherence. So if they come up with some particular aspects that will cause more adherence. So the more adhered they are, the more damage they will get. So let's talk about why does Bacteroides cause such a tissue damage. So let's talk about the virulence factor, because remember, giving antibiotics to these patients may not be enough because you have to deal with the tissue injury that you saw in this person who had a scrotal injury coming up and spreading all over the skin. The most important, again, number one is adhesins. So it has those adhesins which will adhere to peritoneal surfaces more than anybody else. And one of the adhesion is polysaccharide capsule. That's like a sugary, gluey, sticky thing, polysaccharides. It's going to go and attach. Number two, that they have this capability of having fimbriae. So fimbriae are like hair-like projections, and they are extracellular molecules if you pay attention to typical gram-negative, and you will see that they tend to adhere. And also keep in mind in one of the slides that I showed you, especially the tables, especially the pathogenesis lecture, the tables where we talk of adherence, we talk of many other things where particular virulence factors are discussed. One of important thing that I want you to know, what are the bacteria that attach to fibrinogen or fibronectin or any particular sugar molecule expressed on the surface of those epithelial cells. So that's important. Number two is that they are protecting from phagocytosis. And of course, all bacteria that are protecting from phagocytosis have a capsule. So that again, a capsule there. If you look at their short chain fatty acids, especially succinic acid, which is usually produced during anaerobic metabolism. So they don't need air. When they start making up their energy plan without an air, they will produce this short chain fatty acid, especially succinic acid, and that will itself inhibit phagocytosis and intracellular killing. Protection against oxygen toxicity. They have, of course, classical molecules, and I would rather have you make a chart, make sure that you know which bacteria produce catalase, coagulase, and superoxide dismutase, because I did add quite a few questions tomorrow testing you for that particular enzyme, because these are all important. The most important are catalase and superoxide dismutase because they inactivate hydrogen peroxide and other superoxide free radical which are there. So bacteria get phagocytosed by neutrophils, and neutrophils release those oxygen killing. In response to that, they release this enzyme, chew up that oxygen, and then they basically are protected from oxidant radicals. Why do they cause tissue damage? Because they have cytotoxic. You see that kind of tissue damage, cyto means cell, toxic, toxicity. They are very much a feature of gram-negative anaerobes, and this especially are present in both virulent and avirulent. And what do? They cause tissue destruction. They inactivate immunoglobulins, especially on the surface of epithelium. And then again, as we said earlier, they resist oxygen toxicity because they have SOD, superoxide dismutase. So they basically all play an important part. Then if you remember when I talked about Clostridium, I said when there was no treatment for Clostridia, patients used to be kept in oxygen chambers to increase oxygen content to save these patients and kill bacteria. They produce toxins. One of the most important toxins they produce is called heat labile zinc metalloprotease. And also keep in mind, I'm very fond of asking this question. When I say exotoxin, I want you to know whether it's a heat labile or heat stable, because that will tell you whether you're going to get rid of it by boiling or not. So that's a very important point. The other thing is that it has a special metalloproteases. So metalloproteases are scaffolding of the cell. So if cell is round, and this is like scaffolding, if it is destroyed, cell will fall apart. So metalloproteases are important for this one. This toxin particularly causes morphologic changes and F-actin rearrangement. You saw these kind of F-actin in muscles. You see the muscle configuration of actin-myosin. That is the feature of that. So if it is destroyed, then again muscle cannot contract. So if you remember a cell like neutrophils, it has to do diapedes to move forward by changing the configuration of the cytoplasm which is there. And in this case, this toxin will mess up the F-actin, and then you will result losing salt and fluids. Enterotoxin also induces IL-8. IL-8 is notorious. I may have said many a time. Many a bacteria will seem to, just like the criminal calls the police. I mean, there are problems and they call neutrophils for help. But of course, neutrophils are not coming for help. They are coming for tissue damage, because that's what, unfortunately, the flip side of that is. So you will have more information that you need. So one of the important things you have to remember, I said other day as well, that the tissue damage can be from microbe itself. Tissue damage can be from the toxin released by microbe, but tissue damage can also occur from your own host immune response, where it goes out of control, and that happens most of the time. If you look at epidemiology, they are pretty much very fond of being everywhere, especially human body. And then again, the important thing that you saw over here is that one of their function is that they stabilize the resident bacterial flora. I think you must be familiar by now. What do I mean by that? So there is a fixed number of bacteria which are said normal flora. If you give an antibiotic, overzealous use of antibiotic, you mess up the ratio. And then again, one bacteria grows more than the rest of it. So Clostridium difficile, if it's a minor population of normal flora, right, and you give antibiotics, this now doesn't become normal, it overgrows to such an extent that it will cause pseudomembranous colitis. So exactly in the same way this happens, and you can see this will prevent colonization by pathogenic organism from exogenous sources. So if you have all your friends filled in as a normal flora, and there are no seats to occupy for any pathogenic bacteria, it's good for you. If you kill by use of antibiotics your normal flora, you give a chance to those bacteria which are not normal and maybe pathogenic to disturb the ratio. And that again, if you remember, I discussed that as a feature of vaginal flora and the effect of hormone, lactobacilli, and all these things, because that was very important. Now, normal protective organisms produce serious diseases. They are normal. They may, when they move from their endogenous to normally sterile site. That again, concept that we're trying to dig in is that they are your normal flora as long as they don't invade you. They don't go into your system in your normal sterile places like tissues and blood. if there's cut, ulcer, inflammation, immune system goes down, then you give them a chance, and they're not going to be your friend anymore. So they will go for the opportunistic infections. Now, you can see from here, the organism is a resident flora, but able to spread by trauma or disease. So basic concept, pretty much the same, that any normal flora bacteria can become abnormal, though it may not have very strong virulence factors because of increase in number, because of its ability to go and invade a normal sterile tissue or blood. Now, a bacteria which has all these different kinds of virulence factor, what kind of clinical diseases that it can cause? So you can see, let's talk about the one that I showed you as an impact slide. So you see abscesses. So abscesses can be intra-abdominal, it can go into brain, infections, and then again, gynecological infections because they're in abdomen. After hysterectomy, after C-section, once you open the abdomen, and if it's a nick or any kind of a cut or any other thing, they have this ability to come out and attach to your peritoneal cells. And skin and soft tissue infection, you saw that. Some of these bacteria may have minor. Also keep in mind that when I ask you and give you a choice in terms of picking up the common or the most prevalent or the most important clinical disease or a side effect, and your book or table gives you a list. So keep in mind this usually a list in terms of priority, list in terms of prevalence. In this case, respiratory tract infection will be number one. And again, sometimes your book gives 80%, 20%, 30%. So these are percentages wise, right? So gastroenteritis, of course, but it comes in the end, right? It may be one of the features, one of the remote features. So when a person comes with a intra-abdominal infection or gynecological infection, I would rather look for Bacteroides or abscess before even looking for any other microorganism that may cause it. Because right now, I think your mindset is that since we are talking about bacteria, so you're thinking gastroenteritis is only from bacteria, but we haven't started talking about viruses and parasites and fungi. So then you'll get a bigger picture. But also keep in mind, coming back to that impact slide, if you pay attention to the legend there, it's a polymicrobial infection. So that's another important thing, that don't think that you just get hit or infected by one bacteria. You may have more than one, a group of them, polymicrobial. So that's why we have sometimes given you a therapy where we give you more than one antibiotics, a combination of antibiotics. Liver abscess, you can see typical huge liver abscesses. Bacteroides fragilis is known for that. How do you diagnose? Diagnostic slides, I would want you to pay attention because I did ask you some of the diagnosis for tomorrow's exam when I feel that that was important, especially for drug resistance. So I will ask you some of the drug-resistant antibiotics, hoping that that's something that you need to know. We have talked about MRSA, methicillin-resistant Staph aureus. I talked about vancomycin-resistant enterococci. So these was the bare minimum that you need to know. And then also keep in mind, I'm going to ask you that if a particular bacteria is a drug-resistant, what are the choices, alternative choices of antibiotics do you have? So that's something I would expect you to know. So microscopy, culture, biochemical identification, these are three important things. And keep in mind that they have a resistance to kanamycin, vancomycin, and colistin. This is a serious thing. It's highlighted because these are the first line of antibiotics you want to give. And then again, stimulated growth in 20% bile. That's something when we want to grow them in culture. But also keep in mind that something that can grow in bile, it should suggest to you that it's going to cause liver and gallbladder infections as well. Because bile should have been antiseptic for some of the bacteria because of the nature alkalinity of that. Acid should be antibacterial, but those bacteria have mechanisms to survive that kind of high acids or high bases or any of that sort of thing, and they are culprits. Acute salpingitis, which is the infection in the gallbladder. Chronic salpingitis. Cholangitis. Salpingitis basically is tubal uterine tube infection. So cholangitis, not salpingitis. If you want to grow them on bile esculin agar, you can see we have to add bile there because bacteria wants to grow in bile. As simple as that. And you can see a typical structure, and they have a inhibition by bile and gentamicin. So you can see the gentamicin was added to this medium, and basically that over here, when you add gentamicin, they don't grow. So that's also an important thing for you to remember. And then again, some of them may be resistant to gentamicin, and then they will cause more problem. How do you treat them? Antibiotic therapy. When you have a lot of tissue damage, remember the rule of surgery. The rule of surgery is wherever there is a dead tissue, remove it. That's why you see debridement, that you see amputation, you see all these kind of very radical surgery, because if you do not do that, that dead tissue is going to be a culture medium for anaerobic bacteria. They will love it. You don't want that to happen. So it's rather to cut that tissue out. We call debridement. We call surgical intervention, especially if it's a serious anaerobic infections. Then the other important thing is, this is a gram-negative. All members have beta-lactamases. This enzyme renders bacteria. So pretty much we already had a big lab on that, different kind of beta-lactamases, and what do they do? Protein binding, penicillin binding proteins, and so on and so forth. And then again, we already discussed part of normal microbial flora. Endogenous mean your normal flora. Exogenous mean from outside. Most of the time, the problem, if you have for Bacteroides, is from your endogenous flora, your own bacteria, right? And then the problem is that these diseases are virtually impossible to control. So that's another important thing. There are many reasons for that. One of the reasons is that your immune system gets used to giving them a pass, because the immune system cell, dendritic cell, come and sample this bacteria and tend to know them, make friends with them. And all of a sudden, these friends become foes, and then we have a problem. Disruption of the natural barrier around the mucosal surfaces when you are using surgical techniques, diagnostic techniques, when you are poking holes, penetrating scopes, and so on and so forth, you basically will inoculate your external bacteria into the system. So that's, again, like use of even simple syringes. You have to make sure that you take proper precautions. So if you invade the barriers, you may want to have a prophylactic treatment, and antibiotics may be needed. Okay, and I'll jump to the next bacteria, another slide, because I know they're short bacteria, but again, I mean, as I said earlier, those of you who have an access to online book, they will have the liberty and luxury of looking at some of the flashcards there, some of the case studies there, and they have been found to be quite useful. And it depends upon your manner of study. It also depends upon your ability to cram the material. Okay, so let me close that.